Hello and welcome, dear students. Today we are going to discuss about the topic transient polymorphism. The main objectives of today's lecture are to know the basics of genetic polymorphism, to understand the relevance of genetic polymorphism to evolution, to know the concept of transient polymorphism, to understand the evolution of transient polymorphism, and finally to understand various examples of transient polymorphism. Dear students, let's first start with introduction to genetic polymorphism. We know that genes are located on chromosomes and different versions of a gene are called alleles. The standard allele found in a population is referred to as the wild type allele. Most animals and plants have at least 2 copies of each chromosome, one inherited from each parent, that is from mother and father. The copies of the genes found on these homologous chromosomes may be identical for that gene and may carry 2 copies of one allele. In that case, the organism is called homozygous, or the copies of the genes found on these homologous chromosomes may be different, and in that case the organism is called heterozygous. Alleles differ from each other in their sequence of nucleotides on DNA, which may change the structure and function of the protein in gene codes for. Because of this, different alleles may have different effects on an organism's appearance or ability to survive. These effects can be helpful, harmful, or neutral. A polymorphism is the occurrence of 2 or more clearly different phenotypes within the same population of a species. Whereas alleles are the individual components of a gene, polymorphisms are the individual components of a trait. Consequently, polymorphisms reflect the involvement of more than one allele for any given gene in the gene pool. Genetic variation is generated, maintained, and lost in a population by the interplay of various evolutionary forces, including mutation, migration, selection, and random genetic drift. A lox with 2 or more alleles in a population is considered polymorphic. Polymorphisms can be assayed across a range of molecular levels, from changes in single nucleotides to differences in protein structure, that is blood groups, to gross chromosomal differences like inversions. Most studies in ecological genetics begin with one or the other of the 2 most prominent patterns in nature. One is the adaptation, the fit between an organism and its environment, and second, the polymorphism. Polymorphism, that is the maintenance of 2 or more phenotypic or genotypic forms within a single population by natural selection. Although natural selection is the only evolutionary force that can account for adaptation, several evolutionary forces acting alone or in combination can sustain a genetic polymorphism, at least transiently. Dear students, let's now discuss about the significance of genetic polymorphism. Genetic polymorphism is defined as the variations among chromosomes, alleles, or other DNA sequences. Whether genetic polymorphisms are studied in the form of allozymes, RFLP, mini and microsatellite variation, or DNA sequences have become useful tools in a variety of research fields such as population genetics, evolutionary genetics, systematics and molecular phylogeny, human genetics, agricultural genetics, and forensics. Genetic polymorphisms through multiple alleles at individual loci provide a mechanism to tag a gene or a piece of DNA, providing a powerful tool for a variety of investigations. These investigations cover a wide berth of objectives, including identification of genotypes in paternity and forensic studies, tracking the movement of individuals in field studies following the progression of selection experiments in laboratory populations, mapping of quantitative loci affecting economical traits in plants and animals, mapping of disease genes in humans, and evolutionary comparisons of DNA sequences and chromosome organization between related species. The uses of genetic polymorphisms are almost endless. Dear students, let's discuss about the transient polymorphism. Transient and balanced polymorphism are two types of polymorphism that arise due to two alternate alleles of a gene. In transient polymorphism, from the two alternate alleles of a given locus, one allele is progressively replaced by another. In balanced polymorphism, on the other hand, the two alleles are in equilibrium with each other. Both versions of the gene are maintained in the population. Transient and balanced polymorphism can lead to phenotypic change and may alter protein expression. As we know, transient polymorphisms occur when there are two alleles in the gene pool and one allele is gradually replacing the other. This is due to a strong environmental pressure causing directional selection to eliminate one allele from the gene pool. This transient polymorphism is an unbalanced, unstable polymorphism and is not predictive. Dear students, let's understand the transient polymorphism and melanism concept together. A situation when there are two alleles in a gene pool and one allele is gradually replacing another one is called as the transient polymorphism. This is due to a strong environmental selective pressure causing directional selection to eliminate one allele. The peppered moth is an example of evolution in response to environmental change. There are two types of these moths. One species has a light color while the other one is darker. When Britain began industrializing, the soot from the factories would land on trees, and so the darker moths then had an advantage over the light ones as they could easily hide from predators. Before the soot, both types of moths were eaten by predators. However, now that the darker ones were able to hide, the lighter ones got eaten more often. The population of the darker moths rapidly increased while that of the lighter ones rapidly decreased until only the dark moths were left. All the lighter moths were less adapted to the environmental change and so they could no longer survive in that new environment. So this is a classical example of transient polymorphism. Dear students, if a rare gene gains and preserves some overall advantage, it will spread until its former normal allele is reduced to the status of a mutant. While that process is taking place, but not before or after, it will generate a transient polymorphism. Such an occurrence is not possible unless a genetic reconstruction has occurred in the population or it has expanded into a new and different habitat, or else the environment has changed. For the recurrent nature of mutation ensures that any species will have had repeated opportunities for incorporating into its genetic outfit those genes which have proved useful to it in the conditions it has so far experienced. Moreover, transient polymorphism may not always be carried to completion. Before the former rare mutant becomes transformed into the normal allele, its progress through the population may be arrested because it has had the opportunity of acquiring an advantage in the heterozygous state. For example, we can note moths inhabiting industrial regions get blackened by smoke. Moths become more black or blackish in the polluted areas due to the progressive replacement of one allele from the allele responsible for excess melanin production. Therefore, light and dark melanin variant of the moth is a transient polymorphism directed by industrial melanism or directional selection. So, peppered moth is an classical example of transient polymorphism. As we know that transient polymorphism is a situation when there are two alleles in a gene pool and one allele is gradually replacing another. This is due to a strong environmental selective pressure causing directional selection to eliminate one allele. The peppered moth is a classical example of evolution in response to the environmental change as already discussed. Actually, there are two types of these moths. One species has a light color while the other one is darker. When Britain began industrializing, the soot from the factories would land on trees, and so the darker moths then had an advantage over the light ones as they could easily hide from predators. Before the soot, both types of moths were eaten by predators. However, now that the darker ones were able to hide, the lighter ones got eaten more often. The population of the darker moths rapidly increased while that of the lighter ones rapidly decreased until only the darker moths were left. All the lighter moths were less adapted to the environmental change, and so they could no longer survive in that new environment. This is to say, they have all been subject to transient polymorphism. In Britain, where it first appeared, over 100 species have been affected by such industrial melanism. This has made notable progress in other countries also. It was soon reported in the great manufacturing region of northwest Germany, and it now affects a large number of species there. More recently, it seems to have become widespread in the principal centers of industry in Eastern Europe, in Yugoslavia and Czechoslovakia and Poland. The phenomena has attained impressive proportions also in North America. Selection of this kind has doubtless been responsible for the situation recorded in many animals in which darker populations are associated with darker habitats. For instance, Dice and Blossom analyzed colonies of the mouse which inhabits isolated outcrops of rock rising out of the sandy desert of Arizona. These range in shade from a rather pale disintegrated granite to black lava, and in the 18 localities examined, there was a close correlation between the coloring of the mice and that of their background. Dear students, let's now discuss the similarities and differences between transient and balanced polymorphism. Genetic polymorphism is one or two or more variants of a particular DNA. Transient and balanced polymorphism are two types of polymorphism that arise to two alternate alleles of a gene. In transient polymorphism, from the two alternate alleles of a given locus, one allele is progressively replaced by another. In balanced polymorphism, the two alleles are in equilibrium with each other. Both versions of the gene are maintained in the population. Transient and balanced polymorphism are two types of polymorphisms visible in populations. Transient polymorphism refers to the progressive replacement of one allele of a gene by another allele. In contrast, balanced polymorphism refers to maintaining of both two different alleles of a gene over time. So this is the key difference between transient and balanced polymorphism. In other words, one allele is involved in transient polymorphism while both alleles of a gene are involved in the balanced polymorphism. In a nutshell, transient and balanced polymorphism are two types of polymorphisms seen in populations. Both give rise to phenotypic change and affect gene expression. Transient polymorphism takes place when one allele is progressively replaced by another, causing polymorphism. Hence, only one allele is affected in transient polymorphism. In contrast to the transient polymorphism, two different versions of the gene, that is, two different alleles, are maintained over time in balanced polymorphism. Therefore, both types of alleles are maintained. Thus, this is the key difference between transient and balanced polymorphism. Transient and balanced polymorphism can lead to phenotypic change and may alter protein expression. The term balanced polymorphism was coined by one of the prominent ecological geneticists, E.B. Ford, who actually has divided selective polymorphism into two categories, the transient polymorphism and balanced polymorphism. Transient polymorphism caused by a new favorable mutation in the process of displacing its ancestral allele by natural selection was considered unlikely because advantageous genes will usually have been already incorporated into the genetic constitution of the species, whereas balanced polymorphism, on the other hand, is a situation in which two different versions of a gene are maintained in a population of organisms because individuals carrying both versions are better able to survive than those who have two copies of either version alone. Balanced polymorphism is a system of genes in which two alleles are maintained in stable equilibrium because the heterozygote is more fit than either of the homozygotes. Hence, the difference between transient and balanced polymorphism is based on how the alleles behave. So, genetic polymorphism provides us with the ability to predict inter-individual differences in susceptibility to clinical disease. Biomarkers of susceptibility include polymorphisms in drug carcinogen metabolism, in DNA repair capacity, and in genes that control cell growth. Wide variations in drug carcinogen metabolism have been widely investigated and clearly shown to have an important determinant of individual cancer susceptibility and adverse drug reactions. Such polymorphisms in drug carcinogen metabolizing enzymes may be due to heritable and or to environmental factors, and the modern applications of metabolic phenotyping and genotyping methods to epidemiological studies has provided new insights into such gene-environmental interactions. Dear students, it was all about today's lecture on transient polymorphism and its significance. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.